really happy that Jared has agreed to meet with us today. I've been associated with the American Chestnut Foundation for a long time. I've known Jared, or Dr. Westbrook, for several years, and have worked with him on various projects. There's been a huge change in the American Chestnut Foundation's sort of direction. They started by making hybrid trees. They're into the 6th generation on hybrid trees, just essentially crossbreeding Chinese and American, with the goal of achieving a tree that's resistant to the blight and is 95% American chestnut. But as Jared will tell you, there's a group in New York who essentially has come up with a tree that's fairly resistant to the blight and is 99.99999% American chestnut genes, which is pretty remarkable. So Jared, I'll let you tell us a little bit about your background if you like, and then go ahead. Notice that this meeting is being recorded, and we hope that this meeting will be made available to the public. So might be a good idea to mute your screen during the presentation. And with that, Jared, you're on.
Hey everyone, thanks for having me today. I started with the foundation in 2015. It's the beginning of 2015, so now it's almost 6 years since I began. I learned of this job while I was in India practicing yoga, and I realized, and I was doing my PhD at Florida, and I realized that I could live in Asheville, do all the same kind of tools with genetics that I've been doing at Florida, which was working on pine trees, but apply it to conservation, which is a unique application of some of these tools. A lot of the times they're used for agriculture and growing pine trees for plantations. So this is a unique application of biotechnology and breeding to put a tree that was killed by a fungus back out into the landscape, into forest settings, not in monocultures. So I love living here in Asheville now, and the chestnut really represents this place of Western North Carolina, and I really want to spend the rest of my career and life here. So it's also about feeling a place of home, bringing these trees back to our home mountains here. So I'm going to talk to you about, let me share my screen. I'm going to kind of give you a broad overview of, let's see, share and full screen. You guys, what do you see? Do you see the main PowerPoint view, or do you see the weird multiple slide view?
We're seeing both of them.
All right, let me swap displays. How's that?
Perfect.
Yeah.
All right.
Yeah. Ken,
Okay.
you might want to turn your video off.
You want me to turn my video off?
For Ken.
Okay.
Okay, go ahead.
So this picture here, I just wanted to point out to you, is taken on, the picture on the left is taken in on the top of, around Montreat, Lookout Mountain. So these trees, American chestnuts, used to be highly abundant throughout the Western North Carolina mountains. Right now, there's probably about 40 million trees that are remaining in the forest, but they're mostly stump sprouts. And occasionally you see a wild tree that is on top of the mountain, and it is growing temporarily and produces chestnuts and then dies from the blight. But you can see in this other picture, these trees used to be massive, and this is a classic picture from the Smoky Mountains prior to the blight. So they're called the redwoods of the East. So American chestnut now, I mean, if you've driven around the Blue Ridge Parkway, for example, you can see, in Virginia, chestnuts made out of barns made out of chestnut wood, the chestnut rail fences. It used to be just a utilitarian tree to tan leather, things like that. And it's highly rot resistant, lightweight wood. So a lot of woodworkers really love working with chestnuts. In Europe, they fatten pigs in the forests with chestnuts. So it'd be amazing to have this tree back onto the landscape. In 1904, many of you probably know the story, but fungal disease called Cryphonectria parasitica was imported into the United States via bringing Asian chestnut species that were used for nut production into the US. And the Asian trees are been living with the blight for millennia, and they're resistant to the disease, whereas the American chestnuts are naive and they're susceptible. So this disease was introduced at the turn of the century, and then by 1950, it had spread throughout the entire range of American chestnut down into, you know, where the heart of the range is, which is right here in Western North Carolina, and it killed over 4 billion trees. So you can see kind of the extent of the destruction in Shenandoah. This picture, for those on the phone, is a skeletal landscape with lots of all these trees that are standing and dead. You can see it was at certain parts of the landscape highly abundant, especially on the ridge tops. It likes this well-drained mountain soils, and then Shenandoah was highly abundant. And so today, the tree lives as a shadow of its former self. It gets the blight, and the blight attacks the stem. It goes down into the bark of the tree, and it basically strangles the tree. So it's not able to conduct water up to the leaves. Luckily, the nut is able to sprout from the roots. So what we have now, if you walk around and you can identify these trees, is a lot of trees that grow up from the root sprouts, but they have these like dead stems. And so they just kind of are stuck in this cycle of living as root sprouts, but not ever making it to the top of the canopy, never growing up to the top of the canopy. Occasionally, these trees will flower if there's like a gap in the forest, or somebody cut a road, or they built a house. A lot of times we'll be looking around in subdivisions where they're putting houses on the top of mountains, and that's a great place to find flowering chestnuts, because they get light, they start to grow up to the canopy, they get the blight, and they die back. But at a certain point in their life cycle, they're flowering temporarily. So it's functionally extinct, meaning it doesn't reproduce on its own in the wild very often. Very, very few of the trees are reproducing. And the ones that do are isolated from one another, so they're not able to make babies. They need to have another, partner nearby to make seeds. There are a few partially resistant American chestnuts, so it's not like the entire population is uniformly susceptible, but we know of maybe like 20 of these trees. Some of them are in North Carolina. There's one here. This is in Avery County in the center. The tree is maybe 10 inches in diameter. It has some blight on it, but it miraculously has been living with the blight and perhaps survived. So there's been some work with American Chestnut Cooperators Foundation, for example. This is another group working on chestnut restoration, and what they've been doing is breeding these trees together, bringing pollen from one to another because they're isolated from one another, and planting them out and seeing if they have any resistance. And they do have some partial resistance. Is it enough for restoration? Probably not. So in 1980, let me close the door here. In 1983, the American Chestnut Foundation was started with this idea that blight resistance is, like in crop plants, a lot of times, disease resistance is a fairly simple trait, meaning a few genes control the resistance. So with that in mind, what you could do was proposed by one of our founders, Charles Burnham, who was a corn geneticist. You could take a hybrid between the American and Chinese. The Chinese has resistance, but it grows like a short statured tree. So it doesn't necessarily grow to the canopy. Whereas the American tree grows very tall, but it doesn't have any resistance. Well, you could take those hybrids and cross them back to American chestnut over 3 generations and basically dilute out all of the genes from Chinese chestnut, except for those that are involved in blight resistance. So each generation, we make these controlled pollinations between the hybrids. We inoculate them with chestnut blight fungus, and we select the individuals that have the least severe cankers. And then we do this again over successive generations. And basically what we're trying to do is get a tree that grows like American chestnut, but has the resistance of Chinese chestnut. So you can see this, this is a multiple generation process. We're talking trees, so it's been a long time. We're now 37 years into this. And what we've done so far is we've gotten these trees that are, 95% American chestnut. Now we take those trees and we cross them together. So a tree has a chance of getting resistance from both parents. So we have to generate large populations, and it's kind of like a roll of the dice, whether a tree inherits the genes or not. So it's a very laborious and long process. So what do we got in the end? So we planted these seed orchards, which are composed of we have two seed orchards at our main research farms in Southwest Virginia, just like 2 hours north of here. And we have trees that basically have this huge range of resistance in American chestnut ancestry, because it's been actually a little sloppier than we had originally thought in that diagram. So we have, American chestnut as a reference on the left here, and it, gets the blight and dies and resprouts over and over again. And then we have trees that have a spectrum of American chestnut ancestry and resistance, from trees that are like, 93, 95% American chestnut that have partial resistance to blight, to trees that have a little more Chinese chestnut ancestry, up to 50%. And what we found is basically the trees that had more Chinese ancestry, so they inherited more of their genome from Chinese chestnut, less from American chestnut, were the most resistant trees. So this idea of backcrossing, that resistance is actually a simple trait, is suspect. That actually it's a lot more complicated than we had thought. So it's hard, if there's more genes involved, it's harder to get trees that have both the resistance and the American chestnut ancestry. So kind of just a little dive into genetics here. We mapped where in the genome chestnut has 12 chromosomes. We have 23 in humans, chestnut has 12. We mapped where in the genome is the resistance correlated with a Chinese ancestry, okay? And we have these two parents, Clapper and Graves, that we started with. They're kind of like the Adam and Eve of chestnut. And that we found basically regions on 9 of 12 chromosomes that are associated with resistance. So it is suggesting that we have a more complex kind of resistance than we had thought. So that's why we're getting this partial resistance in our breeding program. We actually mapped where in the genome. This is kind of just nerdy, but we'll go into it. Where did the trees inherit Chinese genes, and where did they inherit American genes? And this figure is showing the yellow is they inherited 1 copy of a Chinese gene and 1 copy of an American gene. The blue is they inherited 2 copies of the American gene, and the red they inherited 2 copies of the Chinese gene. And basically what we found across the genome is that these trees inherited mosaics of Chinese and American ancestry. And basically what we have is trees that inherited different genes for resistance. So they have subsets of resistance genes. So we're kind of doing a salvage operation. We're trying to find the best parents in our breeding program that we can continue to breed with. But right now we have a lot of mixed results, I could say, with it. So here comes in with a transgenic tree. These people at New York have been working on putting in genes from other species. This is what transgenic means. It's like trees are, they're putting in genes from, let's say, wheat. They use a bacteria to infect a tissue of chestnut, put this gene in to the chestnut genome, and see if it has an effect on resistance. And William Powell, the researcher at New York who developed these kind of trees, knew that the blight fungus secretes an acid that creates the cankers on the stems that kills the tree. And this gene from wheat, he knew, detoxifies that acid. So it's oxalic acid that's secreted by the fungus, and the gene turns that oxalic acid into carbon dioxide, which is what we breathe out, and hydrogen peroxide. So these are non-toxic products to the tree. And this gene is, it was used from wheat, but it's found in stuff we eat every day: bananas, rice, barley, sorghum, cacao, many more. It's also naturally present in a lot of grasses and other like fungi and everything. So this gene is conserved across the tree of life. And when you put it into American chestnut, you can see the trees that inherited the gene in the middle on this figure they're standing up tall, even though they've been infected with blight. The trees that don't have the gene are dying. They're wilting from blight. And even the Chinese chestnuts a little bit are wilting in this figure. This is seedlings. Take it with a grain of salt. The tree, living in an environment with a lot of stress and having to live for hundreds of years, we still have to test if this gene can, leads to stable long-term resistance. But in these early trials, it looks very promising. So we've gone through now a regulatory review process with the US Department of Agriculture to be able to release these trees into the wild. Right now, we have to have these stringent permits to be able to plant these trees, and we cannot allow them to pollinate other trees nearby. We need to cut them down after a certain time. So what the US Department of Agriculture is looking at is, are these trees going to have some kind of ecological unintended consequence? So at the SUNY ESF, the people that developed these trees looked and they compared the wild type American chestnuts without the gene versus the transgenic trees with the gene. And they looked at what kind of fungi live on the roots, whether bees like to feed on the pollen, whether tadpoles feed on the leaves, the herbivory of caterpillars. They did a lot of the trees decompose, the leaves decompose as similar to the wild type trees. So they compared the wild trees to the transgenic trees and all these traits, and basically they found no differences except for blight resistance. And so they submitted all of that data to the USDA for a petition for non-regulated status. And then recently, like as in October through recently into August through October, actually, there's a public comment period on that petition. So people were able to read that on the Federal Register and then make their comments to see, is there anything missing in this petition that we should be considering? And so they're taking that into account right now. All of the comments, there was about 4,300. About 70% of them were positive, 30% were negative or neutral. So they're taking all of that into account, and then they're going to write an environmental impact statement, maybe in a year or something. They'll write this impact statement and then make a recommendation as to whether or not to deregulate the tree. Now, the Environmental Protection Agency also regulates these trees on the basis that they mitigate the growth of this fungus. So they regulate it similar to as they regulate pesticides, even though the gene doesn't actually kill the fungus. So that is also going to go through a review with the EPA. And then the Food and Drug Administration is also regulating for is there any allergenicity to the nuts, things like that. So they're doing this kind of rigorous evaluation between these three agencies. So it's probably going to take at least 2 to 3 years to get through that whole process to even have a decision from each of those agencies. But in the meantime, we're talking about trees. We got to be thinking beyond our lifetime in a way with this. So we're starting with permits from the USDA. We're starting to cross the transgenic trees, which they're derived from basically one single American chestnut tree from New York has this gene in it, and we can't put clones out on the landscape. So we're going to cross that single tree with a lot of American chestnuts to bring in more diversity so that the tree can be adapted to areas as far south as Mississippi and all the way up into Maine. And one of the advantages of this approach is it's a single gene. So it's easier to select for. 50% of the kids, when you do a cross between a transgenic tree and a non-transgenic tree, they inherit the gene that confers this resistance. We can easily select for it with, we can take the nuts, we can poke a little hole from the nuts pull out the tissue from the nut, put it in a solution. If it turns black, the tree inherited the gene. If it stays clear, they don't have the gene. So it's very simple. Rather than planting like, tens of thousands of trees to select 100, like what we're doing in the breeding program, we can make rapid progress, get a lot more diversity. We got to be worrying a little bit about, like, we have this wide range of a tree, so we got to be making sure we have enough diversity, and the climate is also changing, and it's changed in the last 100 years from when chestnut died of blight. So we need to be thinking, like, can we get enough diversity for this tree to kind of have a leg up and be able to continue to adapt on its own in the wild? One of the things that we can speed this whole process up with, we can't start with just take one tree and cross it with 1,000 trees or something, because it would be like Genghis Khan. Genghis Khan, one dad, many moms. The kids would all have Genghis Khan as their dad and a lot of different moms. Okay, so if the population was only based on the descendants of this one tree, it would be kind of an inbred population because it has this one dad. So we need to basically use a lot of generations of breeding to these wild trees to dilute out the dad genome, which is the original tree we transformed with this oxalate oxidase gene, and bring in more of that the wild tree's genome. So it's going to take some time, and we can speed that up by growing the plants once we make these crosses under a high light treatment, so they flower in their first year of growth, and then we can take that pollen, put it on other wild trees. So we're basically diluting out the original founder tree's genome. So that's the idea. We're taking our original founder tree and we're putting it over 5 generations to wild trees. And we're thinking of doing about 670 trees as our wild tree population we're using for breeding. And we're doing this over 5 generations, basically to get more diversity into the breeding program. We also have our state chapters, which Kent is part of. We have a lot of volunteers that have been working with us for the last 30 years. We've been giving them pollen from our hybrid trees, and they've been crossing onto these wild American chestnuts in their backyard. And they've been planting orchards with these hybrids. So you can see in this map where we've found the wild trees and where the people have done the crosses and started planting these orchards. So we have some representation already of diversity that's in our breeding program. And some of that diversity is, hybrids that don't have very good resistance. But if we cross them with this Darling 58 tree, we could conserve that American chestnut diversity. So they would also be blight resistant. So we've already had people involved in this for a long time, and we already have orchards ready to go for this pollen, including in North and South Carolina. We don't know how much diversity we need. There's like 400 million trees left on the landscape. We're not going to use 400 million trees for breeding. That's impossible. So we want to be able to get a representative sample. So we don't want to get, if we are using 677 trees for our breeding program for this transgenic tree, we don't want to get 677 from Western North Carolina. We want to be able to get representation of all of the climatic zones where chestnut grows, and represent all of the diversity and all of the where the tree has migrated, capturing the original, like the tree used to live in only the southeastern United States when the glaciers were at their maximum, and then it started migrating north. So we got to capture like the areas that are most diverse. So what we're doing is we're sequencing the genomes of 500 American chestnuts, and we're looking at how does DNA variation in these trees relate to climate, where the trees grow, and figuring out in the future how is climate going to change, and matching where the trees are adapted to where we end up planting them. That's the end goal with this. So we've been having people go all over the place and collect leaves for us to get a representative sample that we sequence the genomes for. So that's something we've been doing. We're also trying to conserve wild type trees into into orchard environments where we can access them. Previously, we've been going using, like, we call the utility companies and we say, we know where this tree is that's like right next to the road. Can we use a bucket truck that you're using to fix the utility lines to go up and pollinate that tree? Well, that's kind of laborious. So could we, and it takes a lot of juggling with logistics. So could we actually bring the trees to us? And one way to do that would be to go out into the landscape in the winter time. And this is something you guys could help with potentially, or somebody in Transylvania County, is if we know where an American chestnut tree is, we could take twigs off of that tree and graft them onto a rootstock. So the tree will have it in a pot, and then we can plant that in an orchard. And that would be something we could use for breeding longer term in an orchard environment in one location. And one things we've been doing too is the problem with that approach is the trees die of blight, and they, especially where they're grafted, they get the blight in that graft union. So that's definitely a challenge of this approach. And so what we've been doing is growing those grafted trees under a high light treatment, getting them to flower before they have a time to get the blight. And then we get the pollen, and we can use that for further crosses. So the pollen is not susceptible to blight, but the stem of the tree is. But we also, there's been a lot of work on chestnut. So in Europe, they've been able to keep the European chestnuts alive, because the European chestnuts also are susceptible to blight, with infecting the fungus with a virus, the blight fungus, that makes it less virulent. In the US, that approach doesn't work very well because there's a lot of diversity in the fungus. It was introduced many times from China and other places. And that diversity limits the transmission of this virus between fungal strains. But there's actually been engineered fungal strains that enable transmission of the virus to a lot of different natural strains of the blight. And so we are working with the USDA as well to get permits to use this virus-infected fungus to basically treat cankers that we see in our germplasm orchards of the wild type trees, and keep the trees alive longer. Basically, it's the bottom line. We need to treat every individual tree, so it's not a practical strategy for wide-scale restoration, but it does keep the trees alive longer for breeding. So I'm going to kind of skip over some of this stuff. I just kind of wanted to mention in passing that we do have other strategies besides breeding and biotech, this one gene that we're using. We're also trying to understand in Asian species, what are the genes for resistance? What are the mechanisms? And then we could potentially use, if we find out later that the transgenic trees are not as resistant as we thought, we have other genes in mind that we could potentially put into the American chestnut from Chinese chestnut to enhance its resistance. So we're hedging our bets against the uncertainty, because it takes a long time to learn in tree world about whether something is working. And one of the other things besides chestnut blight, there's another disease, unfortunately, that affects American chestnut, and it kills the trees from the root. So rather than the trees being able to resprout, they die from the roots up. And Kent has helped with this, where we have been screening our hybrid trees for resistance to this root disease, which actually the Chinese chestnut has resistance. Our hybrids also have some resistance to it. It's called Phytophthora cinnamomum, and it lives primarily in the south because the trees or the fungus it lives in the soil, and it's limited in its distribution by cold winter temperatures. So in the south, it's colder or warmer, so that the fungus is able to live, especially down in the lower elevation forests. But as climate's warming, winters are getting warmer, and this disease is spreading. So we are selecting for resistance to this other disease as well. And we have hybrid trees that have that resistance. We can breed them with blight resistant trees to get both types of resistance in the program. So if you ever wanted to come and help us, we screen thousands of trees every year for this root disease. And you can see some trees have this terrible root rotting and some trees have robust roots that are resistant. So this is something we do with the U.S. Forest Service over in Bent Creek. And Kent has helped with us a number of times, and Don Surratt and people from Transylvania County. So basically, this diagram is showing it's gotten a lot more complicated. We're not just talking about breeding anymore. We're talking about bringing in biotechnology. We're talking about conserving a lot of germplasm, talking about selecting for a second pathogen resistance, this root rot, trying to keep our trees alive with viruses that infect the fungus, make it less virulent. So we're bringing in a lot of different tools now that we didn't have in the past, including all this genomic testing and everything. So the mission has gotten more complex. However, there's still a lot of need for people to help us with this mission, like finding trees is one example. We have your smartphone, there's an app called Tree Snap. If you're out walking around and you can identify trees, you can use this app to, put in the location of this tree. The smartphone knows via GPS where it is, and you can tell us, does this tree have blight, and is it flowering, etc. We need people to go out and collect the scion wood, the twigs for conservation. We are looking for places where we can plant for restoration trials with our the most advanced hybrid material with the partial resistance. We have these transgenic trees planted out in a number of places right now to test their long-term resistance. That's a little more involved because it requires a permit, but we still need both the specialists, the people in the science realm, and we need the people that go out and just help us in a lot of things, like with our volunteering to find trees or screen for resistance and things like that. So if you wanted to be involved in that, we certainly have opportunities here. So with that, I'll take any questions.
Okay, great. That was a great presentation. I'm just going to start with a quick question while others sort of contemplate. The only transgenic tree that you seem to have out right now is Darling 58. Are there other versions, other transgenics that Bill Powell is working on that might be used to increase diversity?
Yeah, so okay, there's a number of answers to that question. We have the Darling 58. The reason that has the 58 after the Darling is that the gene, the oxalate oxidase gene that enhances blight resistance, has been inserted number of times. 58 is the 58th time that gene was inserted into American chestnut. Okay, so there's different lines that have the same gene, and we're working on putting that gene into different American chestnut backgrounds. So we're not starting with the single Genghis Khan tree. We're starting with Darling 58 plus maybe Darling 100 and Darling 109 or something, and the other thing we're doing is Bill is putting in genes from Chinese chestnuts that we think are involved in resistance. So genes that inhibit the fungus from degrading the cell walls in the plant tissue, things like that. So we're putting those genes in combination with the oxalic acid oxalate oxidase gene to see if there's a kind of additive effect. But that, the transformation process of putting the gene in and getting plants and then putting them out in the field takes some time. So they're kind of in the process right now of just putting those genes into the tree, but we haven't evaluated their resistance yet.
Jared, are there any groves in Transylvania or Buncombe County that are being tested right now that are already in the ground?
Yeah, there is a number of orchards, like there's one in Henderson County called the Pryor Orchard, and that has basically it's based on trees from all over Western North Carolina. We're crossed with our hybrids from our Meadowview breeding program, so it represents unique diversity. So that is already in the ground. There's a lot of orchards in various stages of breeding. I think there might be like 50 or 60 orchards in Western North Carolina. So yes, and we are actually, we have a farm that we just purchased that is in near Alexander, North Carolina, north of Asheville a little ways. And we have another area that's owned by a video game tycoon named Tim Sweeney, who owns a bunch of property in Carolinas, and that's near Hickory Nut Gorge, near Fairview. And so those are two areas where we're looking to like plant more trees that would be managed a little like with like our staff or with Tim Sweeney's, the video game tycoons people. So we need like people that would be able to kind of maintain an orchard over some, like a decade or something. It's a commitment. But we are looking to get places where we can put the wild type trees and keep those alive and use them for breeding. So that's something that if the trees die, we know they're going to die from the blight, but they'll resprout. So if that's one place that I think a lot of people can get involved in that wouldn't be very high risk, I guess.
Hey,
Jared. Oh, excuse me, I'm sorry. Mike Santucci here, Assistant Forest Supervisor on DuPont State Forest, and also a SUNY ESF alum. Pretty cool. Thank you for sharing that. Interested in supporting this type of thing. So I have a couple questions for you. One, are you looking for any particular site characteristics for these outplantings or these establishments? See if I can, if we can work towards that. I'm also curious if the biotechnology has changed the silvicultural or silvical characteristics of chestnut, if you have enough information on that yet.
Okay, so first question is site characteristics. Well drained, sandy soils, great. Sandbox, really good. And like slightly acidic, 5.5 to 6.5 pH. Yeah, those are the main characteristics, like the bottomland kind of site. The other thing to consider is was it an old field before? It might have Phytophthora. Right. If it was. Right. That's because it's moved around Phytophthora is moved around on crops mainly. How
about sunlight? You had mentioned earlier in your presentation about forested settings. So are you looking for like a reforestation on a clear-cut site, or are you looking for forest settings?
Yeah, so it's like intermediate in shade tolerance. So shelter woods, or kind of something where you have like a release cut a little bit, but they have some shade. I think that's a good silvicultural strategy. Clear cuts, they do fine, but one of the issues is just managing the deer. So we have to have like tree shelters that we put up. In Western North Carolina, it's not as bad as elsewhere, but yeah, like we have to manage the competition a little bit, for a few years. So clear cuts, you get a lot of like the other trees and things that grow up, shrubs that shade out the trees. So you need to manage the competition probably for a few years.
Right.
Yeah. And then now Now, as far as I understand what your second question is about the silvicultural characteristics, the tree, there might be, we don't know this yet, there might be a little bit of a cost to expressing this gene in terms of growth. So the tree is putting in a lot of energy into keeping that gene turned on. And it may be before it. So what we're trying to test right now is that cost might be the highest in the early stages of growth before the tree has blight. But then later on, when the tree has blight and it's able to resist the blight, well, then the trees that don't have any resistance will be hindered in their growth because they have chestnut blight. Whereas this tree might be able to grow, even though there's a cost, there's also a benefit. So is the benefit greater than the cost, basically, in terms of having more resistance enough to where it can grow and compete? So these kind of questions, it would be a lot easier to address them in a forest setting once we have non-regulated status, because right now we have to like cut down the trees and keep track of everything, and the APHIS comes out and inspects us like once a year, and so it is quite a lot of reporting and everything to make sure that the trees don't accidentally escape. That's what they're trying to avoid, right?
Yeah.
But if we have non-regulated stats in a few years, well, and we wanted to grow these out and do a test planting, and DuPont, for example, we could design an experiment where we kind of look at some of the, competitive things, what kind of silvicultural treatments would be best, compare it to the wild type American chestnuts that are susceptible, all that kind of thing, I think would be wonderful to do in a forest setting. And I asked the U.S. Forest Service about this, and they're would be able to do it probably in experimental forests, but with a lot more public input, I guess, from all over the place. They're a little bit more hamstrung, I guess, in their ability to do larger plantings. We need like the private landowners and the people in the smaller jurisdictions, the parks and everything, the states, to be able to do this, to be able to test it out, because it's controversial,
Yeah.
So,
Yeah. Well, thanks, Jared. I don't want to monopolize the conversation here, but I do want to talk further with you. Your timing is really good for what we have going on in the forest, and this fits perfectly within one of the missions on the forest. So I really appreciate the information you gave us today.
yeah.
Jared, are there any mature trees right now that are producing nuts in your groves on private land, or do you have to go on and cut those trees down even on private land before they are able to produce?
Oh yeah, there's like a lot of places where people on private lands let us know that they have a flowering chestnut, American chestnut, and we go and collect the nuts. And if you know of them, we certainly want to go out there and collect seeds, because that's preferable to grafting, because you don't have to mess with the tree getting the blight in the graft union and things like that. If you can find seeds, which is rare, or if the tree is flowering but it doesn't have a partner nearby, we could always put pollen on that, wild pollen from another American chestnut on that tree. So we need to find, we want to particularly to know where the flowering trees are. If you know of people, you could let them know, like to look out for them, and we could give you certainly some materials to help you identify them, to make sure, because we get a lot of people that call us and say, we have this tree in our yard, it's like really big, and we think it might be the final American chestnut that lived or whatever, and what they have is a Chinese chestnut in their yard, or a hybrid. So that's very common to see that in private land. But if you're like in a new development on top of a mountain or something like that, and they don't have planted trees, and there's the chestnut, that's probably an American.
Jared, got a question for you. This is Owen Carson. I do a lot of field work, and I have run across flowering and fruiting American chestnuts in the past. I've noted their locations and provided that information, usually to the conservation organizations that I'm working with. But I mean, if I'm out and I can find, hulls with actual, nuts in them, viable nuts, I mean, should I collect them and mail them to you guys?
Yes. Yes, absolutely. I would contact you go on our website, and there's this link that says, have you found a chestnut, on the front page. And there's it'll tell you like, you can submit this tree locator form with like the coordinates, and you can send a leaf sample. If you find nuts, like certainly collect them.
Okay.
And you can also use Tree Snap. If you're just like out hiking and you find a bunch of trees, and you can just quickly put that into Tree Snap, even if the tree is like, a resprouted thing that's not flowering. Like we want to know where these trees are like growing, and like we're doing climate modeling, like and niche modeling, like where do the trees grow? Where do we find them? So that information is still useful, even if we don't use that tree for breeding or anything.
Okay. And one other question, it's not a follow-up, but I put it in the chat box, but I'm not sure if anyone's looking at that. Have there been any studies? I mean, it's really nice to see all those ecological studies on how the tree performs, in the environment. But have there been any studies on fire adaptation or fire response in the hybrids? Because I'm sure that's going to be probably some component of the landscape, moving into the future as you try to repopulate this thing. And I wonder how it responds to fire.
Well, yeah, it does. Well, the Americans resprout vigorously. The Chinese, the Americans also have these paper thin leaves, so super flammable. The burrs are even more flammable, the part that holds the nut, as compared to Chinese. So the Chinese have leathery leaves, thicker leaves. So I think fire adaptation is part of the American chestnut, how it was able to gain abundance, because it needs like some disturbance to be maintained, because it's not like the most shade tolerant tree. So if there's no disturbance, eventually the more shade tolerant trees probably end up overtaking it. But a lot of the silvicultural stuff was, we have a kind of limited knowledge, because we don't have like blight tolerant trees to be able to look at like forest succession or something like that. So there've been a few studies just looking at like flammability and comparing the Chinese and American, looking at how do the trees like resprout after fire disturbance and things like that. And we know for sure like that disturbance is an important part of maintaining the trees in the landscape.
Thank you.
Other questions from anyone? Okay, go ahead.
I have a question, Kent. This is Tori.
Hey, Tori.
Hey there, Jared. In relation to Owen's seed collection question, is there a specific process to get twigs to you? Any methodology that you could just briefly describe?
Yeah, I can send you a protocol, but we like to make sure that the twig is dormant, first of all, and then you want the most vigorous growing part of the stem. And sometimes the resprouted material is not growing. So there's not like the green growth from the last year that's vigorous. But yeah, the more vigorous that growth is, like if the tree is recently resprouted or something, and it's like putting on new growth like gangbusters in a area that was in a forest gap, that's like the growing tip, the green part is like the best part for grafting.
Yeah, that sounds like basic horticulture, but it's something that I run into a lot more often than flowers and fruit. Thank you.
Yeah. If you're able to find them, and maybe Kent, you can give people my email address. I will send you a protocol. We are looking now, from now until like February, for that cyanwood for grafting, and we're going to graft it at our research farms in Southwest Virginia, all the stuff from the South, starting this spring. We actually have a grant to conserve like 300 plus different wild type sources of American chestnut from across the range in the next few years. So we're certainly looking for, probably like 20 or 30 different trees from Carolinas, especially like in areas that we haven't necessarily represented. And so we have this kind of map where do we have trees that we've conserved, and where do we have gaps in that conservation. And we've been kind of going out to groups and saying, these are the areas where there's gaps, go find this tree, go find us trees and send us twigs. But, you know, any diversity that we get would be still valuable.
Okay, go ahead.
Jared, this is Dave Casey. I just wanted to appreciate, thank you for presenting. I've been part of some outplantings of the B3F3s since 2009, and just to hear, you know, the hope of something that's maybe more effective and more likely to actually grow into the canopy and be real restoration is very encouraging. So thank you very much for everything you're doing. So.
Yeah, I hope that it works too.
Yeah.
It's a big experiment. And you know, the breeding program, I think what I'm trying to tame is it's a kind of a salvage operation. We have stuff that's good in the breeding program, but we have a lot of stuff that's not so good. And we're trying to separate it into two piles now. And that's where we're using the genomic tools to kind of help us pick the best parents and move that ahead. But I'm very happy, you know, excited that we have another tool with biotechnology and we can potentially combine them. So I think we're kind of doing like something like hedging our bets by diversifying our approaches in all of these, you know, different ways. Like I also am not going to totally rely on that transgenic tree until we have long-term resistance data, and we might need the partial resistance from the hybrids combined with that gene. So all of those are on the table. We have a lot of different paths that we can take going forward.
Gotcha. Well, thank you very much.
Anyone else? So I really want to thank Jared for giving us this presentation. It's very informative, and you covered a whole lot of areas. We'll try to hopefully get this recorded presentation available to the public. Go on the website for the American Chestnut Foundation. They have a whole lot of resources that are available there, a lot of information you can download. It's a great story, and if you join them, you'll get a lot of updates, both by newsletters and by email that sort of posts what's going on. So, Jared, I know you got another presentation at 11 o'clock. For those of you who might want to hear more from Jared and have another chance to ask questions, at 11 o'clock they're having a webinar, and you can log in and listen to that as well. Otherwise, Jared, I'm going to let you go. So thank you very much. You're welcome to stay for the business meeting, but you probably have more important things to do.
Well, thank you for the opportunity, y'all. And send me an email if you have any more questions, or if you want to tell me where you found a chestnut. Kent, you can provide my information, and we'll go forward from there. I appreciate your interest. All right, see everyone.